Hey everyone, welcome back. Ready to dive back into the inner workings of our cells. Today, we're zeroing in on protein production. Like, picture a microscopic factory humming away in every single one of your cells.
And just like any factory worth its salt, it's not just about cranking out the product, right?
Yeah.
The cell needs a system, a way to make sure those proteins, those building blocks, are assembled correctly and shipped to the right spot. That's what we're unpacking today.
Right on. And we're focusing on a particular organelle, the endoplasmic reticulum, or ER for short, which is basically where proteins go to get folded, modified, and sorted. It's like the cell's own little assembly and shipping department all rolled into one.
A very good analogy. Your notes hit the nail on the head, by pointing out that not every protein made in the ER is meant for shipping. Some actually get to stick around.
Oh, that's right. It's not a one-size-fits-all situation in there. More like you've got some workers on the factory floor and some products prepped for delivery.
Exactly. The proteins hanging out in the ER lumen, that's like the center of the ER, they're like those essential factory workers, crucial for the ER to function. Then you've got proteins embedded right in the ER membrane, acting kind of like gateways or checkpoints, controlling what goes in and out.
So how does a cell keep those two types of proteins straight? It's not like they're wearing different colored vests in there.
Well, imagine this, all right? A single pass transmembrane protein, the kind stuck in the ER membrane. Picture it like a buoy floating on a lake.
Okay, I can see that. Go on.
Part of it's got to be anchored below the surface, right? While the other part bobs above. These proteins, they've got this special sequence, a stop transfer sequence, and it acts just like that anchor I mentioned. It halts the protein from moving through the membrane, leaving a part sticking out.
Ah, I get it. So the cell's basically sorting proteins from the very beginning, even within the ER itself, making sure everything's in the right spot right from the get-go. That's incredible at such a tiny scale.
It speaks volumes about how organized a cell really is, and this is just the first step on a protein's wild ride.
Okay, so we've got our proteins in their starting positions. What happens next? My notes, they mentioned something called N-linked glycosylation. And I'm not going to lie, it sounds a little intimidating.
It's actually quite elegant when you break it down. Think of it this way, okay? The cell attaches a small sugar tag, it's called an oligosaccharide, to the protein. That's essentially N-linked glycosylation in a nutshell.
A sugar tag? So it's like the cell is slapping a sticky note on the protein or something. But why? What's the point of a sugar tag?
That sugar tag, that's like a shipping label. It carries information, and that information helps direct the protein to its correct spot within the cell.
Okay, now that makes sense, like an address on a package, right?
Yeah.
So without that correct sugar tag, a protein could end up like lost in the cellular mailroom.
Exactly. And what's even more fascinating is that the N-linked glycosylation, it's not a one-and-done deal. It can be modified even more later on in the Golgi apparatus, adds another layer of complexity and fine-tuning to the whole protein trafficking system. But we'll circle back to that.
Wow, it's already clear this is a super organized system, with checks and balances at every turn. But I'm also realizing that even with the right address label, the package itself, the protein, it needs to be sorted correctly in the first place, right?
Absolutely. You've hit the nail on the head there. A protein's shape is critical to its function. If it's not folded correctly, it's like jamming a square peg into a round hole. It's just not going to work.
So how does a cell make sure those proteins are getting folded properly? My notes mention chaperone proteins. They're like the folks on the factory floor making sure everything is in tip-top shape.
That's a great way to put it. Those chaperone proteins, like BP and HSP70, they're essential for protein folding. Imagine, try to fold a complex origami figure, but someone keeps bumping into you chaos. Chaperones, they prevent those bumps. They create a space for the protein to fold the right way.
Like little robotic arms, carefully guiding and assembling the parts.
Precisely. And here's where it gets really interesting, okay? One of those chaperones, B by P, it actually puts the brakes on the folding process a bit.
Wait, slow down. That seems counterintuitive. Wouldn't you want the protein to fold as fast as possible?
You would think so, but rushing things, it leads to errors. It's like trying to cram for a test. You might get some stuff right, but you're going to make mistakes. B by T, it's all about quality control. Gives the protein that extra time to, explore different folds and settle into most stable shape.
So, like, sometimes a slower, more careful approach is actually better for these proteins. It's not always about speed. It's about that precision.
Exactly. But what happens when that factory floor gets slammed? Like super crowded, tons of unfolded proteins piling up. That's when the cell flips the switch on its emergency response system, the unfolded protein response, or the UPR.
The unfolded protein response. Okay, yeah, that sounds kind of intense. What's actually going down in the cell when this UPR kicks in?
Think of it like this. You've got this huge rush of orders at your factory, way more than usual.
Yeah.
The UPR is basically the cell hitting that panic button, like, whoa, hold up, got to get a handle on this.
Okay, I'm with you. So how does the cell actually regain control? Does it just start bouncing orders? Sorry, closed for business.
Actually, it's a two-pronged approach, which is pretty cool.
Right.
First, it'll temporarily ease up on protein production. No sense adding to the backlog if you're already swamped, right?
Right, makes sense.
And second, it cranks up production of those chaperone proteins, those helpful assistants we were talking about.
So it's like calling in extra hands to manage the chaos and prevent a total meltdown.
You got it. Remarkable system, but gotta say, not always foolproof. Sometimes the demand, it can just outstrip even the best efforts, you know?
That's a little worrying. When would that happen?
We'll take type 2 diabetes, for example, which you mentioned in the notes. In this case, the demand for insulin, which, as you know, regulates blood sugar, it's through the roof.
Right, because the body's not responding to the insulin effectively, so the cells just keep trying to make more and more.
Exactly. It piles on the pressure for the cells responsible for insulin production. It's like our factory suddenly having to churn out 10 times the usual amount of one specific product. The system gets strained, and these misfolded insulin proteins, they start piling up.
And that's bad because...
Misfolded proteins, they're not just useless, they can actually be toxic to the cell.
Yeah.
Like having a bunch of faulty parts jamming up the works over time can lead to those insulin-producing cells dying off.
That is alarming. So a system that's designed to protect the cell, it can actually become a liability under pressure, really shows how delicate this whole balance is in there.
Absolutely. And it underscores why understanding this whole intricate process is so important, right?
Right.
So we can find better ways to treat conditions like type 2 diabetes, for example.
Okay, so let's say our proteins, they're folded upright, no distress signals, they're good to go, ready to ship. How's that happen? They don't just, hop on a microscopic train to their destination, do they?
You wish. Cells, they rely on this amazing network of transport highways, mainly the secretory and endocytic pathways and their trusty vehicles, vesicles.
Vesicles, okay, yeah, those ring a bell. They're like those tiny sacs you see in cell diagrams, right? Like little bubbles.
That's them, like those little delivery drones you see zipping around, but, microscopic scale. They butt off from one compartment, gobble up their cargo, then scoot over to fuse with another compartment, deliver their payload.
So sticking with our factory analogy, these vesicles, they're like our fleet of trucks and vans moving stuff between departments.
Spot on. The secretory pathway, that's like sending shipments out from our factory to, say, a distribution center. Proteins headed for the cell membrane or for secretion outside the cell, that's their route. Endocytic pathway, that's for bringing things in, like raw materials, maybe breaking down waste.
So the cell's got its own internal shipping lanes, incoming and outgoing. Amazing.
A beautifully coordinated dance, really. And speaking of choreography, we got to talk about the Golgi apparatus, key player in making sure those deliveries get where they need to go.
Golgi apparatus, okay. Another name I vaguely remember from bio class, but honestly, it's a little fuzzy.
Picture the Golgi as the ultimate sorting center, okay? Like a super efficient mailroom that processes, packages, directs everything to the right recipient.
All right, I'm with you. So our proteins, all folded, ready for action, they arrive at the Golgi. Then what? Does it just slap on a final shipping label and send them off on their merry way?
It's a tad more nuanced than that. The Golgi, it's dynamic, see? Organized into these flattened sacs, or cisternae, constantly being formed on one end and maturing as they move through the Golgi stack.
So not just one big room, it's more like an assembly line of sorts.
Perfect analogy. As proteins move through these different compartments, they're getting modified further. Each cisterna, it has its own set of enzymes, like specialized workers, right? Performing specific tasks, maybe adding or trimming sugars, attaching other molecules, like those final address labels, or even instructions for the protein's final stop.
Like those packages that bounce between processing centers, each step adding another layer of sorting and refinement.
You got it. And just like any good system, Golgi's got quality control too. If a protein takes a wrong turn, ends up in the wrong compartment, needs a little extra work, it can be sent back to the ER, retrograde transport, like that return to sender option on packages,
Right. Okay, so we've got our proteins all boxed up, labeled, ready for the final leg of the journey. But how do they actually get loaded onto the right vesicle? And how do those vesicles know where to go? I imagine you could get a little chaotic in there, all those tiny trucks zipping around. It's like a giant microscopic traffic jam in there. How do these vesicles know which proteins to load up and where to take them?
You're right. It would be a total free-for-all if vesicles just grabbed whatever and zoomed around. That's where these coat proteins we talked about earlier come in. They're like those different containers you'd use for shipping. A big, sturdy crate for heavy machinery
Okay.
versus a padded envelope for something fragile. COP2, COP-I, clathrin, those are all examples. Each forms a distinct vesicle, you see.
So the type of coat protein on the vesicle determines what it can carry and where it's going, like a dedicated delivery service.
Exactly. It's like having different trucks for different routes or delivery types. But it's more than just the trucks themselves, right? Imagine a team of expert logistics coordinators making sure the right packages end up on the right trucks. That's basically what adductor proteins do. They're like the intermediaries. They bind to both the coat proteins on the vesicle and specific receptors on the cargo, making sure everything matches up.
So it's like double checking the address, making sure everything's going to the right place.
Yeah.
Okay, so we've got our proteins all packaged up in their designated vesicles. They're labeled, they're ready to hit the road. But how do they actually get offloaded at the right destination? Do they just like bump into their target and hope for the best?
Not quite. It's a much more controlled process, thankfully. It all starts with docking. That's exactly what it sounds like. The vesicle sidles up to its target compartment and attaches. Picture a ship pulling into harbor. Or even just that satisfying click when you plug in a USB drive.
Ah, gotta have that satisfying click. So no accidental deliveries to the wrong department then. But how does the vesicle know it's docked at the right spot?
That's where tethering proteins come in. Think of them like harbor pilots, recognizing specific markers on the vesicle and guiding it to the right docking site on the target compartment.
Like vesicle carrying insulin over there by the cell membrane, please.
Exactly.
Yeah.
And once that vesicle is all locked in, the next step is fusion. That's where the vesicle membrane actually merges with the target membrane. It's like two soap bubbles becoming one.
So the cargo just smoothly gets incorporated into the target compartment. No rough unloading or anything.
You got it. And this fusion process, it's orchestrated by another set of proteins, snare proteins. Think of them like those specialized couplers that connect train cars. They make sure that a vesicle only fuses with its intended target, preventing any accidental spills or mix-ups.
Wow, so it's not just about getting the cargo to the right place. It's about making sure it arrives intact and in perfect condition. The level of care taken at each stage of the journey is incredible.
Absolutely. And it gets even more mind-blowing, actually. Remember we were talking about a protein's shape being so important to its function. Well, throughout this whole delivery process, from the ER to the Golgi to the final destination, the cell actually maintains the protein's orientation. It doesn't just get dumped haphazardly into the target compartment.
Wow, so they're not just shipping boxes, they're also unpacking and arranging everything at the destination. That is impressive.
It really is a testament to the amazing complexity and efficiency of cellular processes.
Well, we've covered a lot of ground today, haven't we? From the ER to the Golgi, all those busy little vesicles, and of course, the importance of proper folding and delivery. It's been quite a journey. And on that note, to all of you listening, we hope this deep dive has given you a renewed appreciation for the incredible logistics happening inside each and every one of your cells, every single second of every day. It's a microscopic world of constant activity, but also incredible precision. So next time you see a package delivery truck, take a second to think about that amazing journey of a protein, because odds are something very similar is happening inside you right at that moment. Thanks for joining us on The Deep Dive.